In this module, we'll examine neurons. First, we'll consider the structure of neurons and how they operate. We'll then consider how neurons can code and represent the external world. Early neurophysiologists who wanted to study neural tissue would use stains that highlight the cells in our nervous system. These early stains weren't very effective, and if you look at this figure, what appears to be happening is just a lot of strands going every which way with no real start and stop points. They called this a nerve net, a network of continuously interconnected nerve fibers. It wasn't until Raymond y Cagil used a Golgi stain on newborn animals, which gave enough resolution to be able to determine more specifically what the structure of neurons were. A Golgi stain, unlike the stains that had been used, stain only one of every 100 cells. It helps isolate them. Furthermore, by using newborn animals, Cagil was using neural tissue that was less dense. He proposed the neuron doctrine, that in fact our nervous system was made up of non-continuous individual cells that are transmitting signals in the nervous system. This diagram represents what a typical nerve cell would look like. The main body of the cell is located here. The small area inside would be the nucleus of the cell. There are several branches coming out from the cell body. These shorter branches are called dendrites. They're connected to other neurons. They receive the information that other neurons are transmitting. This neuron transmits its information down an axon. This axon is a much longer track. It's myelinated, or we could say insulated, to help improve the efficiency of the firing. The action potential, the direction of information flow, then comes into the cell through the dendrites, then extends down through the axon. At the end of the axon, these terminal buttons here would be connected to the dendrites of a second neuron. The bottom figure is an example of a sensory neuron. Here, there is the addition of a receptor. Our senses all have receptors that transduce physical energy into neural impulses. This particular receptor, a haptic receptor, transforms touch into an electrical impulse. So a stimulus coming in from the environment will cause this receptor to become distorted in shape, which causes an electrical signal to fire and travel down the axon to other neurons. If we look at how neurons are connected, as I said before, the axon of one neuron is connected to the dendrites of another neuron. If we were to blow up where these connection points, what we would find is that the nerves aren't connected directly. There's a very small space between the transmitting cell and the receiving cell. We call this the synapse, or the synaptic gap. The neuron sending the signal does not actually send an electrical impulse across this gap. Rather, it releases chemicals, which has an effect on the receiving neuron that may or may not cause it to want to fire as well. The firing of a neuron travels down the axon. So for example, if we were to take an electrical meter and measure the difference in charge between the inside and the outside of an axon, what we would find is that there is a negative charge overall. It's kind of like a battery, where there's a negative pole and a positive pole, and there's a difference across the battery. If we connect the two ends of the battery, we get a flow of electricity. Here, the inside of the cell is negative relative to the outside. When the nerve impulse begins to travel down this axon, there is a shift, a depolarization that happens where the resting potential climbs sharply and actually for a brief period becomes positively charged. As that impulse continues to travel down, we see the action potential begin to repolarize again and eventually return back to its resting level. Edgar Adrian won a Nobel Prize when he developed this technique of measuring the firing rate of single cells. He connected his monitor to a sensory neuron and then tried 3 different stimuli: light, medium, and heavy pressure. What he found was that the response rate, the number of times the cell fired, was a function of how much pressure was being applied. If the pressure was just light, then the cell would fire fairly infrequently but faster than its normal firing rate. If you applied more pressure, you would get more firing, and heavy pressure caused a maximal amount of firing. So the amount of firing was directly related to the amount of pressure. This was how the sensory neuron was coding that quantitative aspect of the stimulus, the intensity of the stimulus. How do single neurons represent information in our brain? It's first important to consider that our experiences are not direct with the stimuli, but rather our experience with the external world is formed through representations in our brain. We call this the principle of neural representation. Let's consider the phantom limb patient that Balyan Ramachandran talked about in his TED Talk. Here's a patient who's experiencing extreme pain in a limb that doesn't exist. Ronald Melzack, a Canadian researcher at McGill University, an expert on pain research, has described phantom limb as an example of how pain is in your brain. If I have a sore limb, a sore hand, my brain tells me that the pain is located in my hand. But of course, it isn't. What's happening in my hand may be causing the pain, but the actual pain is in my brain. It's my brain's interpretation of the neural signals coming from my hand that lead me to the experience of pain. Because of this, I can have pain when a limb doesn't exist. I can have phantom limb pain because that pain isn't actually in my limb, it's in my brain. We'll look at this more when we look at perception, because all of our perception really follows the same principle of neural representation. While you're listening to my voice and thinking it may be coming from your headphones or a speaker, it isn't. It's actually being internally represented in your head. My voice is coming from your head. One way neurons could represent information would be to have neurons that fire when certain things are being presented to it. Probably the simplest example is research that was done by Hubel and Wiesel. Hubel and Wiesel won a Nobel Prize for this research. They were looking at the primary visual cortex of cats. This is the first area visual information gets processed at. And they were presenting different stimuli, oriented bars, or oriented bars that moved, or shorter bars that were moving, and so forth. And we're looking for cells in the visual cortex that would respond to these different stimuli. And what they found was that there were specific cells that were finely tuned to a particular orientation, other cells that were finely tuned to a particular orientation for a bar that was moving. Some were tuned to short bars, some were tuned to long bars. But these single neurons were quite specific in the type of stimulus they were looking for. They needed to see a bar at the particular orientation moving in a particular direction in order to fire. Complex stimuli might be represented in a similar way, and in fact there is some evidence for that. Gross and his colleagues in the early 70s were doing single-cell recordings from a monkey's cortex, and they presented the monkey with different stimuli. The particular neurons that they were measuring from seemed to be specifically tuned to hands. So when presented with these range of stimuli presented here, what they found was that areas in the temporal lobe that they were measuring from, they couldn't find cells that really wanted to react to 1, 2, or even 3. As you moved further up and started to develop images that became more hand-like, they found cells that would fire more and more rapidly, particularly for the last 2, 5 and 6. Here there was maximal responding of these cells. It was as though these cells were specifically tuned to recognize hands. They found similar cells that were tuned to faces. Rolls and Tovey, examining single-cell recordings, discovered that certain cells would fire much more rapidly when presented with faces, as we see in the top here, than if they were presented with images that did not contain faces. When we talk about sensory coding, what we're really talking about is how neurons represent the characteristics of our environment. We have these cognitive representations, and we know that the cognitive representations are somehow working on a neuronal level. So the area of sensory coding is quite critical to understanding how we represent the external world. What we've seen so far are single cells that seem to respond to different stimuli. So one way in which our neurons might code the external environment is through something called specificity coding. Here an object is represented by the firing of a specialized neuron that only it responds to. The idea is sometimes called a grandmother neuron. It's the idea that when you see your grandmother, you have one specific neuron that fires that tells you this is your grandmother. So for example, here when I see Bill, neuron number 4 becomes active. That's Bill's neuron. Mary, neuron number 5 becomes active because that's Mary's neuron. And then Raphael, when I see Raphael, neuron 6 becomes active. So each specific person has a specific neuron tied to them. There's probably a problem with this type of coding. What if that neuron dies? Neurons don't always live forever. If Bill's neuron dies, I think that would mean that I wouldn't recognize Bill anymore. And we don't usually find neuropsychological cases of people who lose the ability to recognize some people. We might get poorer at facial recognition as we get older and our memory begins to fade, but we're not going to lose the ability to recognize specific people. You're still going to recognize your grandmother, even if your grandmother neuron dies. So another possibility is population coding. Here we can take an object and have it represented by a pattern of firing over a large number of neurons. So here, when I see Bill, I have some neurons like neuron 3 and 8 that are very active, other neurons that aren't very active like 7 and 10, but overall I have this particular pattern over the 10 neurons that is specific to Bill. If I look at the pattern of neuronal firing I have when I look at Mary, I can see that the pattern is very different. Here for Mary, 5, 6, and 7 are really quite active, whereas they weren't very active for Bill, and 2, 3, and 4 aren't very active where they were active for Bill. And then if I look at the pattern for Raphael, I get a different pattern again. And so you can imagine that just using 10 neurons and the pattern of activation across those 10 neurons, we could actually come up with a great number of patterns to recognize many different faces. Another type of coding, similar to population coding, is sparse coding. In sparse coding, we're looking at representation here in a pattern of firing for a smaller number of neurons. So for example, now we can see that for Bill, neurons 2, 3, 4, and 7 are important. For Mary, it's 4, 6, and 7. For Raphael, it's 1, 2, and 4. We still have different patterns, but it's a matter of some neurons being active for Bill's face, and some neurons being active for Mary's face, and some neurons being active for Raphael's face, and the rest of the neurons being more or less inactive. I've included a reading with this section that talks about the grandmother neuron again, except this time we'll take your textbook's name and we'll call it a Steve Carroll neuron. Researchers found, while looking at single-cell recordings, several neurons that seemed to be very specifically tuned to Steve Carroll's face. They also found neurons that were tuned to Jennifer Aniston and other famous actors and actresses. But if we look at the firing rates of these neurons, we can see that there's several of them in this range here that are very, very active whenever they're looking at Steve Carroll pictures. Does that mean that they are Steve Carroll neurons? This seems to be evidence for the sparse coding. We could claim that, oh, I have a Steve Carroll neuron, and It's just like my grandmother neuron, and it fires whenever I see Steve Carroll. But more than likely, these cells are part of sparse coding. So these cells do respond specifically to Steve Carroll, but there's a number of cells involved responding to Steve Carroll. And there might be other people that they respond to as well. It's not possible to exhaustively test every possible face that you can present to these stimuli, and it probably is an example of sparse coding. In our next video lecture, we'll look at how our cerebral cortex is organized.